Imagine a key that doesn't just fail to unlock a door, but instead melts into the lock, permanently jamming it shut. This is, in a way, what a molecule called lenacapavir does to HIV. The target isn't a lock, but something far more intricate: the virus's own protective shell, its capsid. For decades, the fight against HIV focused on the machinery it uses to copy itself. But what if you could simply break the container it carries that machinery in? Or, more accurately, make that container so robust, so unbreakably stable, that it can never open to do its job? The HIV capsid is a beautiful, conical structure, almost like a microscopic spaceship. It's assembled from hundreds of copies of a single protein, known as P24. These proteins link together in a precise lattice of hexagons and pentagons, forming a sealed vessel. Inside this vessel is the virus's most precious cargo, its RNA genome and the enzymes needed to hijack a human cell. The capsid's job is twofold. First, it must protect this cargo as it travels through the hostile environment of the cell's cytoplasm. Then, upon reaching its destination, the nucleus, it must perfectly disassemble, releasing its contents at exactly the right time and place. It's a delicate balance of strength and designed fragility. Lenacapavir exploits this balance with ruthless efficiency. It's a capsid inhibitor. When the drug molecule enters an infected cell, it finds these P24 proteins. It binds to a specific pocket where two P24 proteins meet, acting like a molecular staple. This has two devastating consequences for the virus. First, for incoming viruses, the drug latches onto their capsids and makes them too stable. The shell that was supposed to gently open at the nucleus is now fused shut. The viral RNA is trapped inside a useless protein cage. Second, for a cell already infected and trying to produce new virus particles, lenacapavir interferes with the assembly process. It causes the P24 proteins to clump together incorrectly, building malformed, non-functional capsids. The new viruses that bud off from the cell are duds, unable to infect anything else. What makes this approach revolutionary isn't just its clever mechanism, but its persistence. An injection of lenacapavir can suppress the virus for 6 months. How? The drug is administered in a special formulation that, once under the skin, forms a small concentrated deposit, a depot. From this depot, the lenacapavir molecules seep out into the bloodstream incredibly slowly, but consistently. Think of it less like a flood and more like a steady, microscopic drip. This is possible because the molecule is astonishingly potent. It works at picomolar concentrations, meaning only a tiny amount is needed to be effective. This slow, sustained release maintains that perfect therapeutic level for half a year, a stark contrast to the daily peaks and troughs of a pill. The secret to this incredible potency lies in shape. The interface between two P24 capsid proteins forms a three-dimensional pocket with a very specific geometry. Decades of research allowed scientists to map this pocket down to the individual atom. Lenacapavir was then designed, through computational chemistry, to be the perfect molecular key for this very specific lock. Its own shape allows it to slide into that pocket and form strong, stable bonds. This is not a loose fit, it's a high-affinity interaction. This precise molecular handshake is why it doesn't interfere with our own cellular proteins and why so little of it is needed to do its job. It only fits where it can cause the most damage to the virus. This level of specificity represents a fundamental shift. For years, HIV treatments targeted the virus's enzymes, the workers. Lenacapavir targets the virus's architecture, its very structure. It's like stopping an army not by fighting its soldiers, but by dissolving the armor they wear and the vehicles they travel in. This opens a new front in the war against HIV, providing a powerful option against viral strains that have developed resistance to older drugs. It's a victory for structural biology, proving that by understanding the precise shape of our enemies on a molecular level, we can design weapons with unprecedented accuracy, turning a virus's own structure into its ultimate vulnerability.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.